dignitaries and participants to our distinguished lecture series. Today we have with us Dr. Chandrama Saha, who is the President of Indian National Academy. I welcome you, ma'am, and I welcome all the other dignitaries which are associated with us at the present, including Professor Krishan Lal, Professor Sopti, Professor may I to formally welcome all the dignitaries and participants. Sir. Kindly unmute. Sir, kindly unmute yours.
Am I audible now?
Yes, sir.
Audible now?
Yes, sir.
Esteemed Professor Krishnalji, Chief Guest Professor Chandrama Sahu, President Indian Science Academy, Dr. Professor R.C. Sopti, former Vice Chancellor Punjab University, all the dignitaries and the participants. It gives me a great pleasure and it's honor for the GGDSD College Society to welcome you all on this seminar. GGDSD College Society runs 4 degree colleges and business schools, and its premier flagship college, GGDSD College Chandigarh, Sector 32, is A+ NAAC accredited, recognized as a star college by GST, and has both 4 research centers bestowed by the Punjab University, namely in biotechnology, chemistry, physics, and management. We are passing through a very difficult period. For the last 1.5 years, the world has got disrupted because of the pandemic. It goes to the credit of the scientific society that they have shown the way to the humanity how to tackle this, and with the innovation and to face is required. And this scientific
friends, this is the 18th webinar in the last 10 months, which we have been holding as the distinguished seminars under the forum, the Chandigarh Forum for Science and Technology Communication, as well as the Chandigarh chapter of the Indian Science Congress. But this time, Dr. Bachawat has been very kind to join us in this very venture of distinguished lectures, which are going to continue probably for the time to come. Thanks to the Indian Science Congress for allowing us to carry on these activities. Friends, we have very distinguished scientists amongst us. Dr. Krishan Lalji, the former president of the Indian National Science Academy, and the current president, Dr. Chandrama Saha of the Indian National Science Academy. Beside, I can see the fellows of the Academy, Dr. R.K. Kohli over here, Dr. Deka is there, and many other I can see over here, and some of them I cannot see. So I welcome you all to this very webinar, which will be an eye-opener for all of us, and for youngsters, it will give a spark. Let me introduce the speaker of today, Dr. Chandima Shah, is a biologist and president of the Indian National Science Academy. She is also the former director of the National Institute of Immunology, New Delhi. Her research interest centers around the elucidation of the processes that influence cell death programs under varying physiological conditions in diverse organisms. She graduated from Calcutta University and did postdoctoral studies at the Kansas University Medical Center and Biomedical Center of the Population Council at the Rockefeller University, USA. She is an elected fellow of the World Academy of Sciences and fellow of 3 Science Academies of India. She served as vice president of the International Affairs of the Indian National Science Academy and also as a member in the council of 3 national academies. And notable awards include the Ranbaxy Science Foundation Award for Basic Sciences, the J.C. Bose Fellowship, Shanti Swarup Bhatnagar Medal of INSA, Om Prakash Bhasin Award, Archana Sharma Memorial Award, Darsha Ranganathan Memorial Award, Chandrakala Hora Memorial Medal, and Shakuntala Amit Chand Prize, besides many other awards she has got in the life. And she is doing a wonderful job in the academy and taking the academy ahead in the new directions which were started by the previous presidents, including our Dr. Shri Krishan Lalji. I know I have worked with him for some time and found that how dynamic he is. And in fact, I have always been directed by Dr. Krishan Lal not to talk about him, but to the youngsters, a few words I think he will allow me to speak about him. He is an honorary professor, Indian Institute of Technology, Kanpur, and honorary professor of life, the University of Delhi, and visiting professor in our university. He was IBM Indian Fellow at Watson Research Center, New York, visiting professor at the University of Tokyo Technical University, Darmstadt, and senior visiting scientist and physicalist technician Bundestadt, Duisburg. He was visiting professor at IIT Delhi and Jamia Millia Islamia, and adjunct professor at IIT Kharagpur. Also served as treasurer INSA, member advisory board CSIR, director CEL Shahibad, chairman research advisor. So many things he has done over here, and he has held very important positions. Dr. Krishan has made significant contributions in research and development, scientific leadership, and developing international collaboration. Dr. Lal's research work on lattice imperfections in crystals, X-ray diffraction physics, and crystal growth has led to deeper understanding of the nature of real materials and their interaction with radiation and with the external fields. Dr. Lal's students and collaborators had organized 2 international conferences in his honor, sponsored by International Union of Crystallography. Also, Indian Crystallography Association honored him on the occasion of 70th birthday. Dr. Lal has been chairman of 6 international symposia, workshops, schools. He has delivered more than 100 invited talks in reputed national and international conferences, has edited 9 books, volumes, published so many invited papers in journals, chapters, and books, more than maybe 2 century research papers. He referred journals, and he has many patents to his credit. He has a whole 2 honorary doctorate degrees, including 1 from Russian Academy of Sciences, and is very actively involved in the inter-academy programs. He had been vice president of that also, and a co-chair of those programs, and many positions he's been holding. And I'm very grateful to Dr. Krishan Lalji for accepting our request to chair a very prestigious, distinguished lecture by Chandima Shah. I welcome Dr. Ramesh Dekaji who had been a director of the AIMS and done wonderful job over there, and is very actively involved in academics at this moment. Also, the moment he gets our information, he joins our programs, and I welcome Dr. Kohli for joining this very program, and Dr. Shri Lakshman Das Shinglaji, I can see over here, and especially I welcome our Dr. Bachawat, who has joined with this very program on Distinguished Lecture Series of Dr. Chandrama Shah. And now over to Dr. Shah. People are waiting to listen to her deliberations. Dr. Shah, welcome, please. Dr. Krishnalal will preside over this very lecture.
Thank you very much, Professor Sobti, for the kind introduction. I must say that it's a pleasure to be here, and I must thank you for this opportunity. I would also like to thank Professor Krishnalal for agreeing to preside over this. Also now Dr. Bachawat has agreed to be a part of this. I would also like to thank him, and my greetings to all faculty members and students of the Biotechnology Department of Punjab University. Greetings to the members of Indian Science Congress, INSA Local Chapter, and Chandigarh Forum for Science and Technology, and GGDSD Education Society. So I'm grateful to all of them for joining in this. I would like to talk today about. I'm also seeing many people I know. Dr. Kohli is there, and some of them I can't see. But greetings to all of you. Now I will put up my presentation now. Yes, is it visible now? Is it changing?
Visible.
Is it changing? Is it changing?
You click on the left sidebar.
Yeah, I will click on the left sidebar. So yes. So now changing?
Yes, it's changed.
Are you seeing full screen or are you seeing half screen?
Half screen sort of. It's okay.
Okay.
Now it's better, yeah. Now it's better.
Okay. So infectious diseases have been evolving since life on Earth began. And their developments have been influenced by splitting of the continents, the evolution of early primates into present-day forms, and migrations of man around the world, the ending of ice age with warmer temperatures coming in, the invention of agriculture, the domestication of animals, and finally the urbanization of societies. And we must also say the industrialization that we faced. So today I'm going to give some of the basic facts, knowing that there would be students in the audience. So I would seek indulgence from scientists, senior scientists who are here. In early times, the germ theory evolved, and around the Middle Ages, even there was a concept of germ theory. And Ibn Sina, who was a polymath, Persian polymath, he sort of alluded to a germ theory, and he is regarded as one of the most significant physicians of that time. Now, the most important discovery that happened that made a critical contribution towards development of germ theory was the development of microscope by Antony van Leeuwenhoek. And this microscope was a small contraption with a very, very tiny lens here, by which he could see a world that nobody has seen before. And Antony van Leeuwenhoek was a draper. He was not a scientist by profession. And his discovery by a draper, made a revolution in science and changed everything that we looked into. So the germ theory took its actual shape with Louis Pasteur and Robert Koch. There are others also. But the main architect was both Louis Pasteur and Robert Koch. Pasteur is regarded as one of the fathers of the germ theory of diseases, and his many experiments showed that by stopping germs, you can cure diseases. A German physician microbiologist was the discoverer of specific causative agents of deadly infectious diseases, including tuberculosis, cholera, anthrax. And he is regarded as one of the main founders of bacteriology. Now we know that by germ, we refer to viruses, bacteria, protozoa, fungi, and even substances like, even entities like prions. So as germ theory evolved, the question was why everyone is not getting sick, why some people fall sick. So the study of immunity began. So Russian scientist Elie Mechnikov, in the early 19th century, he actually discovered what is an immune reaction for the first time. What he did was he took a starfish larva and put a rose thorn into it. And what he saw was the influx of cells going in towards that thorn and trying to sort of push it out. And this was the first demonstration of an immune reaction. Now, when we can get infected by the oral route through aerosol, through insect bites, through things getting into orally inside our digestive tract. So if this is a picture of an insect bite, where you see that in the blood pool that forms below the dermis, there are various types of immune cells that have moved in. Reaction that Mechnikov saw many years back, and now we know clearly what kind of cells are going in and what kind of reactions are taking place. Now, the three main cells, Mechnikov worked for many years on macrophages, had several elegant papers on this. And the other two who are the sentinels of the immune system in a way are the neutrophils, and they are the first line actually, and then dendritic cells, who are antigen presenting cells. So when we look at immunity now, the lymphoid cells played a very big role in immunity came much later than when Mechnikov worked. So there are two parts of our immunity, and which is very important today. And people, I find, talking about immunity, very naive people who have not been exposed to science, even they are talking about immunity. And so I thought I should present what actually immunity means. So there are two layers of immunity. One is innate immunity, and the other is adaptive immunity. Innate immunity is non-specific, rapid, and immediate memory. And it contains many cells, of which the most important is the dendritic cell, the macrophage, the neutrophil, who have the capability to engulf pathogens, and the other cells like natural killer cells, basophils, eosinophils, granulocytes are there. And when we get a blood report of our count, we see the counts of all these cells that the pathology laboratory actually counts. So the adaptive immunity takes about 1 to 2 weeks. So what happens is that either the macrophage or the neutrophil engulfs the pathogens and dendritic cells even, and digests the pathogens and exposes their parts on their surfaces. And by that way, they can stimulate the T cells, who will differentiate it into helper T cells and killer T cells. And the helper T cells will tell the B cells to make antibodies, and here all the memory B cells are generated. So this is how, in a very, very simplistic term, our immunity works. But the working of the immune system is extremely complex, and we keep on getting surprises that we are getting now in this pandemic. During human evolution, as humans evolved and societies were formed, the hunter-gatherers appeared, so the various kinds of germs started infecting humans. And so there was a tussle between the host and the pathogen. This is true with other animals and animals also, but I'm just talking about humans here. So there was a tussle between the host and the pathogen. And this constant battle for host to evade infections and the pathogens to create infections led to the refinement of many pathways in both organisms. Natural selection worked on such events, and selective forces acting on both the function. For example, The pathogen's desire to survive within the host, selective forces worked on that. And the host's intention of eliminating pathogens, selective forces worked on there also. Selection will actually favor whatever level of virulence maximizes the rate of replication and transmission, but not host mortality. On the contrary, natural selection will favor host defense mechanisms to eliminate pathogens for its own fitness. So this optimum virulence depends on the functional relationship between a pathogen's transmissibility and its effect on host mortality. Now the interaction during the evolution that has come, and we are also evolving. Right now, what we are thinking is, does our immune system have the time to evolve according to the onslaught of the virus that we are actually facing? Since the virus can multiply many times and have many generations going, it can evolve faster than our immune system does. So it's a challenge for us. And the interaction between host and the pathogen during evolution has really shaped the defense system of pathogens and the immune system of humans. Now, I just wanted to give a very, a good example of how some mutations are favored. And I chose the SARS-CoV-2 virus because that is the important thing going around. Although I'm going to speak about most of our work on a protozoal pathogen, but I thought I should take this example where there was an aspartic acid residue D at position 614 of the viral spike protein. And by April of 2020, a single mutation in the genome was found that converted this aspartic acid to glycine. Now, presently, the 614D variant has been all but replaced by the 641G variant globally. Why? Because it provides some selective advantage to the virus. So this G transmits more rapidly than the D mutant, and the number of functional spikes in each of the viral particles becomes 5 times more with this mutation. And so naturally transmissibility and infectivity is more. Patients infected with this 614G variant shows higher viral loads in their upper airways than did patients for 614D. So you can see here that how selective pressures actually retain the favorable mutations. Now, you see, if you look at this graph, this shows that in 2020, from February to June, most of the virus that we saw were the variant that we found was the G variant that we found, and it spread across the globe and kept infecting at a very speedy manner. Now, if we see the surging cases of COVID-19, further mutants like the B.1.618 was a variant that was present in West Bengal. But the 617 variant, which was present in Maharashtra, actually had some advantage in its transmissibility. So now in West Bengal, it has almost replaced the 618 variant. So this again to show that the favorable variants with greater transmissibility and greater infectivity, therefore, are actually retained. But it is not always the case that they are only retained and no mutations are actually thrown off. But there are mutations that are thrown off. So in this also we have seen the infection, the variant in the minks, which is called cluster 5 variant. And this variant in the minks were actually present, and that infected about 10 humans. And finally, there was a huge killing of mink population all over the world, particularly in Denmark, where it surfaced. And now that mutation and that particular variant is almost extinct. One can never tell for sure, but right now they say that it is got extinct because it had some disadvantages. So this is to say that how these this advantageous mutations are kept and disadvantageous mutations are actually waned off. So coming to how the pathogens are actually encountered. So it is a cell-eat cell in the sense that the pathogens are picked up by macrophages. They are put in very specialized chambers called phagosomes. And these phagosomes contain an enzyme called NADPH oxidase, which converts oxygen to superoxide, and that superoxide dismutates to hydrogen peroxide, which forms the hydroxyl radical, the peroxyl radical, and in combination with nitric oxide, the peroxynitride. This H2O2 can, under myeloperoxidase action, can also become hypochlorous acid. So there is a very, very high degree of reactive oxygen species in this particular compartment of the phagosome. Later, the phagosome actually fuses with the lysosome, and the hydrolytic enzymes of the lysosome dismantles the pathogen, and the pathogen is actually killed. So now the question is whether if this fails, because there are many pathogens who have devised ways to stop these fusions, etc. So now this compartment of the phagolysosome, which actually happened after the lysosome fused with the phagosome, what we see is that there are only two pathogens in the world. One is the Leishmania donovani, which I'm going to speak about, which we have worked with, and where it survives in this very low pH kind of conditions inside the phagolysosomes with all these hydrolases and all these reactive oxygen species surrounding it. And another is Coxiella burneti, which causes Q fever in humans and veterinary animals, and they also survive. However, even though they survive in phagolysosomes, both pathogens have very distinct microRNA and mRNA profile expressing in the cells. So, so now, many years ago, actually, when the first dyes became available for showing reactive oxygen species, this is a Leishmania infected cell where we have shown that this is a lysosomal marker, this is a reactive oxygen species marker, and you see that the phagolysosomes containing the Leishmania are actually fluorescing with reactive oxygen species, which meant that there are really an intense amount of reactive oxygen species in these bodies. So now to talk a little bit about Leishmania, Leishmania is actually a digenetic parasite, which is carried by the sandfly. The sandfly takes it up from humans, and then it injects these parasites into another human, where they form rounded forms called amastigotes, and these are the promastigotes which stay in the gut of the sandfly, and they are free swimming forms, and they eventually infect again the human. There could be an intermediate host of dogs also carry Leishmaniasis, but not so much as in our country. So this cycle goes on. So if you look at amastigotes, they're on the scanning electron micrograph. These are the rounded forms, and these are the tailed forms here. And this is how they survive within a phagolysosome. There may be solitary phagolysosomes, or there may be a group of phagolysosomes. So this is the Leishmania, how it looks under in vitro conditions. If we put them in our petri dishes or 24-well plates, they will move like this. So this is about Leishmania, and it is a very, very ancient parasite because it originated in the Mesozoic era when dinosaurs reigned, and the dinosaurs showed the presence of sorrow Leishmania that has been, by forensic studies, it has been found that they contain sorrow Leishmania. It was called sorrow Leishmania. They are found in the Cretaceous Burmese amber 100 million years ago, and Dominican amber, which was there 20 to 30 million years ago. So they're very ancient parasites. Now, the Leishmania first came to India. I mean, it was first discovered here that when a mysterious fever in the 1824 and onwards, that the villages after villages were actually getting wiped off by a mysterious fever. And the patients were very emaciated, they had enlarged spleen and eventually died of the fever, perhaps due to severe anemia because of the spleen association. Now, the discovery of this pathogen was ordered, this was ordered by the British government at that time to the military personnel who were present, the doctors who were present in the military, to find out what was the organism that was causing this disease. So, two gentlemen, William Leishman and Charles Donovan, actually found some structures which looked like parasites in the slides they prepared from people dying from soldiers dying from that fever, or experimental rats injected with the serum of those infected people. And they discovered it, and so there was a problem with the nomenclature. So finally, Sir Ronald Ross stepped in and he named this particular parasite as Leishmania donovani, naming it after Charles Donovan and William Leishman. This disease was actually killing thousands and thousands of people, and it went on up and down. In the 1924 or around that time, when Upgradation prepared a formulation which was pentavalent antimony, and this pentavalent antimony given to this was later realized, not at that time, that when pentavalent antimony is converted to trivalent antimony inside the macrophages, and they go into the phagolysosome and kills the amastigotes. Now, if you look at this graph from Assam, this discovery of his, where he made the pentavalent antimony in a special way, there were thousands and thousands of people who were saved. Now, we should remember that at that time, villages did not have millions of people. They were actually populated less, but there was such a huge problem that villages were getting wiped off. But his invention actually saved many, many people. We used this particular antimony drug at a time when a lot of resistance has occurred to this drug in the field, but we took these lysosomes and we showed that there are actually this antimony also kills by generation of reactive oxygen species. So we have many data this way or that way about the effect of antimony causing increase in reactive oxygen species and killing the pathogens. And when they cannot kill, they do not generate enough reactive oxygen species. So the Leishmaniasis is actually, there are about 300,000 estimated cases annually. Now these diseases in the backdrop of this coronavirus infection have become, people are not willing to think about these, but these are also personal tragedies that keep on happening. So the cutaneous leishmaniasis also is a form which causes huge disfigurement in people, and this happens within the people that are very poor. They have a poor immune system, and that is where it actually affects. And over 1 billion people in the endemic areas are at risk of infection. So there is visceral leishmaniasis, which actually kills people. Cutaneous leishmaniasis causes skin lesions. And PKDL is after the visceral leishmaniasis, there is a cutaneous form which appears, and the cutaneous also has two forms, the localized cutaneous and diffuse cutaneous leishmaniasis. So this disease actually in our country is endemic in Bihar, but it is now surfacing in other parts of the country. Whether that is due to the vector, which I said that sandfly carries this parasite, due to the difference in vector distribution or any other reason, we do not know. So these parasites survive in such a harsh environment. It was known that the lipophosphotican it is a entity which is present on these parasites, on the membrane of the parasites, multi-chain structure, and they protect the parasites from the reactive oxygen species. we showed that in addition to this LPG, ergosterol, which is expressed in a large amount on the Leishmania membrane, actually provides another layer of protection. So these are natural structural molecules that are coming and helping survival of these parasites. So when we generated a knockout where ergosterol would be less, we knocked out this cytochrome P5122A1 gene, and these parasites, low ergosterol, were very susceptible to the drug, the potassium antimony tartrate. We took parasites which were actually resistant to antimony, and we used tyrol biosynthesis inhibitors to treat them, and they became susceptible to the drug after the treatment. So which meant that ergosterol played a very important role in providing protection to the parasites. Now, this much of protection only would not have been sufficient. So we found, and it was reported earlier, that they express two kinds of enzymes, cytosolic tripyridoxine peroxidase and mitochondrial tripyridoxine peroxidase, which actually were protective enzymes, and they cleared up peroxides, which were the products of the reactive oxygen species exposure. And this thione peroxidase system, the tripyridoxine peroxidases were the terminal peroxidase converting the peroxides. So they existed as decamer. This was modeled by the PDB data that was available. And the enzyme cascade here is very, very important. And it contains trypanothione, which is a specialized thiol for this particular parasite. What we showed is that this particular enzyme is present, actually works where it is present, in the sense that the mitochondrial reactive oxygen species are taken care of by the mitochondrial TX and PX, and the cytosolic one by the reactive oxygen species, which are causing damage coming in from outside and killing and destroying the organelles. If we overexpressed the cytosolic tripyridoxine peroxidase, it actually enabled the macrophages to have increased infection, in the sense that there will be 32 parasites per macrophage in comparison to the normal cells, which will only have vectors. There would be about 9.5. So the cytosolic tripyridoxine peroxidase actually helps the parasites to establish the infection. So this is a very important observation, that the localization of these two enzymes are very, very critical. Now, this particular cytosolic tripyridoxine peroxidase has some other function, where I'm going to come in a minute, because it's kind of a multifunctional protein. So this actually applies to all pathogens. The survival within the host is due to preventing oxidative stress-induced killing by the pathogen through manipulation of host housekeeping processes like autophagy, like apoptosis, and through manipulation of host cellular machinery by derailing the signaling pathways in the host, and the pathogen survival is there. Now, when a cell, the first attempt is to kill the pathogen, and the cell is okay. Now, when the host cell is unable to kill the pathogen, it commits suicide or undergoes apoptosis, so that it can take the pathogen with it, and it can get degraded in the lysosomes of the phagocytic cells. Now, subversion of host cell death is a way by which the pathogens actually survive inside the phages. Now, this is very different in virus, thereby reducing mortality of the host cell. So CTX and PX, or cytosolic tripyridoxine peroxidase, has this very important function also to, on one hand, to eliminate peroxides, on the other hand, to reduce the cell death of the host. So this is how the Leishmania parasite has evolved. Now, this is not the only pathway. Some of you may have the idea of the BCL2 family of protein, where it is actually either a anti-apoptotic proteins, which are pro-survival proteins, one may say, and then there are family of proteins which are actually anti-survival proteins. So naturally, there is the pathogens are interested in these proteins. And what we found is that when the Leishmania parasite binds to the TLA2 receptor on the surface through its lipophosphoglicands on its own surface, it phosphorylates the PI3 kinase, the ERK kinase, and the STAT3, which finally leads to an increase in the BCL2 protein, which is the pro-survival protein. And what the BCL2 does? The BCL2 prevents nitric oxide generation by interfering with the iNOS, which is responsible for synthesizing nitric oxide. And in another way, it also phosphorylates AKT and degrades BAD, which is a pro-death protein. So here is a protein which is pro-survival protein, which is increased. It inhibits nitric oxide, and the production of nitric oxide is actually a key defense mechanism against pathogens. It is a pro-apoptotic protein, pro-death protein is destroyed, and that is how the survival of the host cell is ensured. So these are the two examples that I gave you, and it is true with many pathogens. Now, I showed that when bacteria are engulfed, they go into phagosomes and eventually phagolysosomes, and they're destroyed. There is another pathway which deals with the pathogens, which is the autophagy pathway, where a vesicle is formed, and this vesicle inside the cells upon the infection, and this vesicle it picks up organellar products, the misfolded proteins, and as well as pathogens, and this autophagosome form fuses with the lysosome, and there is a degradation of the pathogens. Now, so this is a favorite pathway for the pathogens to target. So if we look at the some of the pathogens, there are many: SARS-CoV-2, Staphylococcus aureus, Salmonella typhimurium, or Mycobacterium tuberculosis. They actually prevent the docking and fusion of the autophagosome to lysosome, and hence prevent their own degradation. True in this particularly with the Mycobacterium tuberculosis, this actually prevents the fusion of lysosome and phagosome, and tries to survive there by escaping also to the cytosol as well. So now what we did was we tried to decipher the role of autophagy in this Leishmania infection, and what we found that the neutrophils, which first pick up the parasites at the infection site, picks up the parasites and starts showing the eat me signals on them. Signals. The macrophage eat them up, seeing those signals, not knowing that they have parasites hiding inside them. And the parasites are then released. While the neutrophil is degraded, the parasites then survive within the macrophages inside the phagocyte. So this is a process by which the transmission of the parasites take place, and this is called the Trojan horse mechanism. If we remember the story of Trojan horse that happened in Troy, where the soldiers came inside the horses as a gift, and then they didn't know that the soldiers were coming in. So it's a similar kind of story that happens here. This is a confocal micrograph which shows that the infected neutrophils here. Now, what we showed is that when autophagy occurs, these red dots are autophagy, there is a high degree of pickup by the macrophages of the neutrophils. So they are picking up the neutrophils with the parasites when autophagy is high. Now, if we stopped autophagy by using an autophagy inhibitor, 3-methyladenine, what we found was that the 3-methyladenine was actually reducing the pickup of the neutrophils. So that is a potential, potential use this autophagy inhibitor. And now subsequently we actually built a model in which we showed that both canonical and non-canonical autophagy was responsible for picking up these neutrophils and causing an infection. So now there is these housekeeping processes I talked about are very important for in pathogens as well as apoptosis and autophagy. So if you look at this slide, you'll see that we have used drugs to kill the pathogens inside the macrophages. And these green dots are actually showing the cells dying by the very regulated process of apoptosis, the pathogens, the Leishmania pathogens. And these are just control cells which are not treated with drugs. So they undergo apoptosis within the macrophages, not disturbing the macrophage nuclei, which are actually red. So which shows that there is a very controlled regulation of this process of apoptosis within the macrophages when you attempt to kill them by drugs or by any other means. So, and this depends on calcium influx, and these are the staining for the calcium, as you can see, and they kill. This process is initiated with influx of the calcium induced by the antimony drug. The single this Leishmania is one of the earliest mitochondrial eukaryotes. It has a single mitochondrion, and this mitochondrion is, if you look at this picture, these are viable mitochondria. These are mitochondria that are switching off gradually. They have a heterogeneous pattern. The green is parts are dead. The red are parts which are still active. This is important because in higher organisms, what happens is that groups of mitochondria are shut off, and the cell has enough energy to die by apoptosis because some groups of mitochondria are still performing. So here, the single mitochondria actually switches off by stages in a heterogeneous manner and retains the capability of these parasites to undergo apoptosis. That is also very important for the parasite for survival. And this autophagy is also essential for survival of this particular parasite because autophagy increases in response to stress, and if you block the autophagy, it results in low parasite viability. We designed an ATG8 knockout, which is an essential gene for autophagy, and we showed that ATG knockout actually interfered with differentiation to infected forms. So this is also very, very important observation where these parasites are actually sensitive to knockout of the ATG8. So pathogens, if we can learn from this narrative, the pathogens are at the wound milieu at the dermis. So the natural oxidants that we have, that what we eat, and we generate, we have oxidants to kill the pathogens that come in. They're not sufficient. Sometimes that we get the diseases. So at the point of pathogen entry, autophagy inducers may help, and there are some autophagy inhibitors also help at some point of time. So both autophagy inducers and autophagy inhibitors are in clinical trial in cancer cases. And BH3 mimetics, which means the BCL2 mimetics, they can work on pathogens inside when they have got inside the phagocytes. And defensive enzyme antagonists, who also can work on pathogen inside phagolysosome. Remember when I talked about the cytosolic triperodoxin peroxidase and the mitochondrial triperodoxin peroxidase. So this is a scenario that is true for all pathogens. The viral pathogens, as I said, they're a little different in their way of causing disease. They can very well mimic many of the host proteins. And that's a very, very interesting area. Now, if we look at the conclusion that we can come to is that the diseases can be prevented by direct intervention like vaccination, which we are realizing now in this huge pandemic, and also reducing pathogens in the environment and by drugs as well, and isolation of infected individuals. That has, we have now realized in a very in a big way actually. And it happened when smallpox used to occur. The isolation of infected individuals is very important, and the treatment of infected individuals, of course, and then education to promote behavior change. And that is a very difficult thing to bring about, promoting the behavior change, as we can see now that hardly, many people are not wearing masks, many people are not maintaining their COVID-appropriate behaviors. So these are the important points that we need to ponder about. So I just wanted to say this much about the Leishmania parasite, along with some other pathogens. Pathogens do survive by thwarting the processes of the host, how they are evolved, and how we should be treating them. Now, one point about Leishmania parasite is that although it is endemic in Bihar and appearing in other places with reduced immunity due to HIV and all, there has been dedicated, very, very dedicated workers in the field who have led to elimination of Leishmania. It's not completely eliminated, and it can come back at any time, but these are very dedicated workers that have worked. And in that note, I would also like to appreciate the efforts that are being done now by the frontline medical workers to deal with COVID-19. So this work was entirely done, whatever I mentioned, at the National Institute of Immunology in New Delhi, where I was for many years. And these are the people who have gone through my laboratory. And these are the funding agencies that have funded my laboratory over the years. Thank you very much.
May I request Dr. Kishanalji to please give his presidential remarks. Dr. Unmute. Unmute.
Thank you, Mr. Sobati, and thank you, Professor Shah. It was a wonderful lecture. You gave a very exhaustive description of this field, which is very, very prominent these days. And because it needs all the knowledge that we have to deal with it, because India in recent past has suffered a great deal from this. I make few comments because you are also somehow linked with being a Chandigarh chapter of ISCA. I want to mention that the Indian Science Congress Association has given birth to many institutions, which also included the science academies, in which we had great dominant figures like C.V. Raman, Meghnad Saha, and so on. And even all the three academies in some way or the other have their origin in this. I also want to recall that the session of Science Congress in Jammu under the presidentship of Professor Sobti, at that time it so happened that our Prime Minister was so busy, he had not created, he had created a committee to look after the affairs of, though normally Prime Minister is the president of the main body, and I happened to be one of the members, and Sobti Sahab was the president of that conference. And he made one demand. He keeps on making demands on me from time to time. He made one demand that in the opening session, there should be a Nobel laureate. Then I have to call upon a friend from Taiwan, and he kindly agreed, and we had that in this. Very recently, he was organizing this Punjab Academy of Sciences. He said, no, koi gadbad ho rahi hai. Then Professor Deka, I think he is also present here, Jagannathan, and many others we included. From time to time, he orders do this or that, and I can't say no.
Request.
So, ये तो मैं just to make it a little humorous. Also, I would like to say one remark about INSA is that one of during my presidency, many new things were done, but one of the things which perhaps even the presidents have not fully, impact has not gone, that every year there is a G science meeting in the world. G science meeting, these are the G8, now G7 countries, plus 5, including India. They hold a summit. And this G Science Summit means primarily it is the G8 countries at that time, now it is G7. So the second one which I attended as a president, the president of French Academy in one of the tea intervals asked me, so about you hosting the next G Science meeting. And this science meeting is important because the statements passed by these science meetings, they go to the main political meeting where they set the scientific agenda. And I had participated in France, and they took us all to Professor Sarkozy, who was their president, and in their palace, etc., etc. So he asked me, how about you hosting the meeting? No, we are not G8 country. So I gave him, it will be great honor, this, that, but I did not commit. And he understood it. So next day, when final day was there, he asked me at tea interval, have you thought about it? Melka, it will be great honor, but I will not propose it. Understand? If I propose, you have an objection. I said, I will. And everybody clapped. So we held one G science meeting outside of G8 countries, which is the only G8 meetings, G science meeting outside of G8 countries till now, as far as I know. And the statements, 2 statements were finalized. And in this also interesting thing happened that the vice president of US Academy who was present, he said that do you want the signature? Because this statements are signed by presidents of all academies, these 13 academies. And I said, yes. He said, I can get you a letter from US Academy president that he endorses and so on. There are all the 13 signatures should be there. Then he said, it will take 2, 3. And he has become my lifelong friend even now. So they also signed, and this signed statement brought INSA at a very higher life. But now also INSA is doing very, very well. And you saw also what a distinguished scientist is there as president. And we are very proud that this is the first lady president of INSA. Though our number of, we hope that the fraction of fellows, lady fellows, increases. At one time, I held a meeting of Asian academies at INSA, and they were counting how many ladies are there in different academies. And to my surprise, the best figure came from Turkey. So they told us it is the 90 years of this reformation in Turkey which has led to this situation. They had the best, even as far as I know, perhaps they are the best figure in the whole of world. And I have later on also twice, thrice they invited me. It's very impressive. Many of them wanted also to have long interaction with it. But presently, my duty is as a co-chairman of the IEP, the Inter-Academy Partnership, which is basically a organization of 110 science academies, which now has joined also with Professor Shah might be knowing, with the Inter-Academy Medical Partnership. So 30, 40 medical academies are also into it. India now, once in a while, they keep on talking to me. I also once addressed them in Delhi in the Medical Association that they should form an academy and become part of the international system. So we have about 113 science and 30 in medical and engineering. So total is now, partnership is about 140. And in this period, we have very much occupied with the COVID-19. We have issued many statements. We also issue communiques and press releases. And from time to time, for example, The European Union had an organization as to how science and society. So they had published a very long interview. I gave about 30, 40 pages. They had touched all, two of us, I a co-chairman and the other co-chairman, two of us gave this thing. Now this is, now we also, I want to mention one where our INSA was also seriously involved. We also pursue projects like the German government had sanctioned 1 million euros for a project on food, nutrition, in which for Asia I was the coordinator, and a collected volume. They are very well received now, are everywhere. We also had from India also not only the Agriculture Academy but the Council of Agricultural Research very seriously involved with this. And then I did not realize that right in the neighborhood of NPL there is also a demonstration, this precision agriculture. It was given by friends. So I said, I was saying, because we got this input from others, but this will finally go into this thing. More recently, about something I think it will interest many people, I came across a World Academy of Arts and Science and Arts and Sciences, and they also have a session every 2-3 years. But in this, the amazing thing is they have also people connected with UN, connected with the UN bodies like WHO and so on. So I participated in one session, and now one message is emerging that if we have to solve the problem of the world, people earlier thought that with UN, formation of UN after the Second World War, it will take care of anything. For health, WHO was there, for trade, etc., WTO was there. But now UNDP and so on, many bodies are there, but now the realization from many of them had served in UN capacities, they were repeatedly mentioning perhaps the present structure will not serve to solve the, in which it is not only political and the other people who are doing, but in sciences, the professionals also, sciences, engineering and others should be involved so that the problems of the world can be taken care of. So I am mentioning this because Sir Sopti and others, they should now think we can even organize a thinking session. Is it worthwhile, or the position very good and altulate the or of GGDSD college or our Sopti is very bigger after this thing, which came giving such professionally technical lecture which is up to date. This only one more sentence I would like to say, that right now IAP also has a group of experts. Group has been formed, about 50 members, and from India, Professor Gagandeep Kang, a very reputed lady, is one of the 3 co-chairs of this group. I was very happy to push her name. And also, so this is a group which continuously is giving this thing. Lastly, I would like to say that IAP supports very strongly the Global Young Academy. In many of our activities, now we are changing some part of our or to bring them formally into this system. But whenever we have a major meeting, IAP invites GYA. Their office was given, in fact, the German Leopoldina, and invited me to go to their GYA. It is very great pleasure to interact with with these young scientists and so on. So I really thank Professor Chandrima Shah for her outstanding lecture and leadership of Indian National Science Academy. And I also thank the college, GGDSD college, for hosting this meeting, and of course the leadership of Professor Sopti there. I also think I saw Professor Kohli also somewhere is there. I had been interacting, and perhaps Professor Deka is also around somewhere. So thank you very much, and I wish you more success, and perhaps will give you more little suggestion to do more for science and technology, because Chandigarh is a very special place, has very active, also the medical part is very strong there. So I wish you all success, and this has been it.
Let me tell you, Dr. Krishan Lal is the co-chair of Inter Academy program. That's why he has given us all the thing. And he is very kind all the time. Before I request Dr. Bachawat to thanks, I must say that we have with us Dr. Anirudh Joshi, companion of the Education Society, and he is always encouraging us to hold all these functions in his college. Request Dr. Bachawat to please report of.
Yeah, sure. Thank you very much, Professor Sopti. It's a pleasure to, first of all, thank Professor Chandrima Saha for this excellent talk. I mean, she really built up this, the story from starting from the history to, and also, getting into COVID and telling us, how even COVID virus evades its post-immune response. It was very fine finding she's made with this very unusual organism. Excellent talk, I think, very nicely tailored for students. For only Professor Sopti can manage to get such luminaries like you and Professor Krishan Lal. I'm also very grateful for Professor Krishan Lal for chairing this meeting and to sharing some of this words of wisdom. He told us the program, about these the things that the Inter Academy is doing. So it was really nice, and also suggestions as to what we can do in terms of brainstorming, etc., etc. So thank you very much, Professor Krishan Lal, also for joining, for chairing this. I'd like to thank, of course, Professor Sopti, who has really been the one who's actually managed to get all the right people to get to for this, for this occasion. So thank you, Professor Sopti, and also Shri Sharma for actually, hosting this, the GGDSD college actually hosting this, lecture at your college. It's wonderful. I've been to SD College many times. We have a wonderful department of biotechnology, and so it's wonderful that you are again hosting such a talk of such a luminary speakers, as well as with the chair like Professor Krishna. I'd also like to thank I can see Professor Kohli and Professor Deka for being there, and also the other co-organizers of Dr. Sopti, Dr. Vipin Sopti of the Chandigarh Forum for Science and Technology, then the Indian Science Congress Associate Chapter, and finally I would like to thank Dr. Navneet Patra, a very, very dynamic young faculty of SD College, who's really always willing to take a lot of effort and running around, and he's the one to actually eventually get this function going. So with these big words, I would like to thank you all for being here for this function. Thank you very much.
Before we close, next week we have 2 lectures on COVID, Monday and Tuesday. Different aspects of COVID will be there. One major is on this black fungus just coming up, and about that is a lecture. And one will be on use of physiotherapy in the COVID treatment. Thank you very much.
Thank you.
Thank you, Dr.
Thank you.
Okay.
Good luck.
I'll only give it to you. Thank you, Dr. So kind.
Thank you.
Thank you,
Kohli
Dr.
sir.
Batra.
Thank you so much. Thanks a lot.
Thank you very much.
Thank you.
Thank you very much.
madam.
Thank you. Thank you.
Thank you very much,
Thank you, Navneet.
Dr.
Thank you.
Chandrabaji. Beautiful lecture.
Wonderful.
Thank you.
Wonderful. Thank you, Roxanne.
Thank you, sir.
Hmm.